Hello, everyone, and welcome to BrainMap. We're so happy to see many people here and to have our speaker here today, but I will let my colleague, who's it? Sergio Osorio to do the introduction. But I just want to thank everyone and then just remind everyone that the BrainMap Seminar is co-sponsored by the P41 grant at the Center for Mesoscale Mapping, which is housed here at Martinos. So with that, I'll just hand it over to Sergio. And I just want to also mention this, maybe that this is a kind of a co-event between the MEG Seminar Series and the BrainMap Series. So if you are interested in the MEG Seminar Series, reach out to me or to Sergio and Jasmine directly. So with that, I'll just hand it over to you. Thank you so much, Sergio.
Hi, everyone. So it is a great pleasure for me to introduce our speaker today, as if he needs an introduction. But anyway, here we have Professor Sylvain Baillet. We are very happy to have him over. We feel like very lucky to have the opportunity to have listened to him yesterday at the McGill and now today here at the Martinos. Sylvain is graduated from the École Normale Supérieure de Paris-Saclay in applied physics and obtained his PhD summa cum laude in physics from University of Paris-Saclay. He has an international career profile, having held positions in France, the USA, and Canada. And he was a Lavoisier Research Associate at the Signal and Image Processing Institute of the University of Southern California before joining the Centre National de la Recherche Scientifique in France. Sorry about that. Code switching here. As a tenure principal investigator in 2000. He became the head of the brain imaging group at the Cognitive Neuroscience and Brain Imaging SNRS laboratory in 2005 at the La Salpêtrière University Hospital in Paris. In 2008, he took a position as Associate Professor of Neurology and Biophysics at the Medical College of Wisconsin and became the founding scientific director of the MEG program at Fletcher Hospital in Milwaukee. He joined McGill University and the Montreal Neurological Institute in 2011, where he has been the founding director of the MEG unit. He was director of the McConnell Brain Imaging Center at the MNI between 2013 and 2017, one of the largest brain imaging research platforms worldwide. Sylvain is the Tier 1 Canada Research Chair of Neural Dynamics of Brain Systems and has been Associate Dean of the Research at McGill's Faculty of Medicine and Health Sciences since 2019. He is also a founding member of the Standing Committee on Science of the Canadian Institutes of Health Research and was elected chair of the Organization for Human Brain Mapping in September 2023. So without further ado, I would like to share the floor with Sylvain. Thank you very much.
Thank you, Sergio. Thank you so much. Thank you, Abbas, also for hosting and for the opportunity to come back at the Martinos with an institution that has done so much and continues to do so much for the development and the growth of brain imaging. Don't know if I need that anyway. And thank you all for being here today. It's very appreciated. I understand it's a special opportunistic kind of scheduling for your BrainMap series, and I'm very fortunate to have you in the audience today. So let me try to get rid of this here. Okay, it's all good. So I don't know, some of you I know I've seen yesterday at the McGovern and were kind enough to attend my lecture. And today, rather than repeating, I thought I would share some very recent data that we have obtained in the lab around the idea of developing or extracting from brain activity a form of brain fingerprinting. So some of you may be familiar with this notion already by the pioneering work of Emily Finn and others in fMRI. And we were curious to see whether we could take this idea a bit further and differently with electrophysiology and neurophysiological recordings, with MEG in particular. So the idea is actually, whenever you do electrophysiology, you face this tremendous amount of data that unfolds over time, millisecond by millisecond. And sometimes very salient features pop out like here in this relatively rare recording of a seizure in the MEG from a pediatric patient we've seen at the MNI at McGill. There is an evident feature, right, that looks very much like a seizure that pops out of, that unfolds over time, and that's really characteristic of the patient's condition. So that's for patients. But more often than not, when you actually collect data for cognitive neuroscience, neuropsychological studies or purposes, well, you see this kind of calm chaos that is unfolding in the raw recordings with no, with the naked eye. It's very rare to spot anything but, anything very salient, except obviously for artifacts of different kinds. But as far as the actual brain activity is concerned, it's very hard to, integrate space and time in your head, even if you're an expert, to extract like a signature of that brain activity of that person, basically. So the idea of brain fingerprinting is actually to, use this time-resolved capability of MEG source mapping and also EEG source mapping to actually determine to what extent it makes us unique, or actually how it contributes to our apparent sense of uniqueness in the world among others, or kinness with others, So this is just an illustration here of the same data I was showing you in the previous slide, which is in real time, 60 seconds in that person's brain activity during task-free. And obviously it looks very busy. It looks all over the place. And yet, resting state analysis of brain networks in fMRI and also MEG have shown that there is structure in that apparent chaos. So does that structure form a form of fingerprint? And how is that fingerprint specific of an individual? And how does it relate to behavioral traits and personality traits? And more interestingly, as far as I'm concerned, if I am a patient, how different is my fingerprint for my age, my peers in terms of age and biological sex and other variables. So that's the motivation behind what we're trying to do here. It's not just a fun thing to explore, although it is, but it's also something that might be meaningfully, become a meaningful tool eventually, hopefully. So I'm going to reuse just one slide from yesterday, and this is the theoretical and foundational, if you will, concepts that we are trying to implement in the lab. We're not the only ones to entertain the idea that the brain instantiates, predictions, cannot reframe, but actually trying to predict what's going to happen to us at every moment. It's what we've done, what we do is actually very much aligned with a long tradition in philosophy and neuropsychology that views the brain as a predictive machine, so to speak. But it's important to actually try to bring together different aspects of neurophysiology and align what they tell us with measurements like MEG and EEG with respect to these conceptual foundations. So the idea is that we are looking at the brain as a set of networks that is hierarchically organized with primary network layers and deeper layers that are interconnected. And as you know very well, these interconnections, they go both ways. And actually, more often than not in the brain, there's a great deal of re-entries from higher order regions, deeper regions, back into more superficial regions. So think as the blue blob as a primary sensory region for instance, the visual cortex, and frontal or prefrontal regions elsewhere, with either direct connections or a certain but few number hubs, cortical hubs, or deeper structures like the thalamus that act as relays. And there are many reasons why these relays are important. Among many reasons, it's also a way to actually ensure that there is long-range synchronization between distant regions with very precise phase locking. But I'm getting distracted here. So the idea conceptually is that the blue highways from the primary to the deeper layers of the network are the bottom up, and the green ones from the deeper layers back to the more superficial one are channels of top-down information. And in the view of the brain as a predictive machine or predicting organ, the green top-down highways convey contextual predictions so that the primary regions, the most superficial layers, at every given time, compute a form of mismatch between the predictions and the actual input from the physical world. And depending on the amplitude of these errors, the magnitude of these errors, we may want to change our behavior to achieve a certain goal or get a certain reward. And if our internal models, internal representations that issue these predictions are wrong, meaning we cannot achieve our goal, well, we have to learn. And this is essentially the procedure. I mean, the idea that these deeper representations are the ones that get updated whenever the mismatch is significant. So these are very generic ideas that are not novel, but the idea is now to look at the pathways neurophysiologically, or the mechanisms, if you will, at the macroscopic level that might implement this form of predictive processing. And what we propose is that at every local region, at every regional level, so more at the mesoscopic scale, if you look at a given circuit of cells, basically they can be broken into more excitatory or more inhibitory cells with different intrinsic dynamics. But what emerges from these circuits, big and small, is that they have the ability to oscillate. So they have a preferred oscillation frequency that is represented in red here, that can be adjusted depending on the state of the circuit and its inputs. And what's also quite fascinating is that on top of this slow oscillation, you get bursts of faster activity, typically in the human brain, in the gamma band, that tend to aggregate at certain phases of the slow cycle. So the slow cycle is a proxy measure of net inhibition excitation into the local circuit. And the blue activity reflects a form of probably local computation in those circuits. And we're talking slow activity from delta to alpha rhythms in the human brain. And like I said, the faster activity would aggregate around the gamma and higher frequency range. So this is a model that we can consider being distributed across many different brain regions and possibly across the entire cortex as a seminal Lego block for local circuit architectures of cortical computing. They are interconnected, as I said before. And what's missing from the spectral picture, if I may say so, is the beta band, which is also a very prominent beta frequency band of the electrophysiological spectrum in the human brain. And what we think is happening, and the possible role for beta bands, is that its activity is organized also in bursts, like gamma activity, but relatively independent from the slow fluctuations of the local circuits. And they are there to basically modulate, like I said, the frequency, the phase of the red oscillation, maybe of the occurrence of when the gamma bursts occur along the slow cycle, etc. So there is ample empirical evidence today that phase amplitude coupling, this form of coupling between the fast and the slow oscillations locally exist in the human brain. The first demonstration was about 18 years ago now by Bob Knight and Ryan Canolty in intracranial recordings of epilepsy patients. You see these alternations of higher, lower, high frequency power with slow oscillations that co-localize at the recording site. So it's an evidence of cross-frequency coupling. And we generalized these observations in non-invasive recordings with MEG source mapping in the resting state with Esther Florin a few years ago before she took her position in Düsseldorf as a professor. And what we did also with Esther is derive the statistics of the fluctuations of phase amplitude coupling between regions, because they do vary over time. And these very slow fluctuations actually we found were a signature of the typical resting state networks that are so recurrently observed in fMRI. And actually, when you look at the spectral properties of these slow fluctuations of the coupling, they actually they peak at a fraction of a hertz very much like the bold fluctuations in the human brain. And there is plenty of evidence now from our lab and others that this schema of bottom-up versus top-down, channeled by different mechanisms of cross-frequency coupling versus beta band, is indeed expressed during also task. And this is an example we published with Benjamin Morillon when he was a postdoc with us. Now he's a PI in Marseille, in France, where during temporal attention, you have to pay, for instance, here, attention to a complex sequence of tones, and in the middle that are presented at the 3 Hz pace. And inside that scrambled sequence of tones, there is a structured sequence to which you need to pay attention, which is presented every 1.5 Hz. So it's a difficult task. And what we found is there's a directed connectivity at 3 Hz, so relatively slow at the presentation rate, basically, of the auditory sequence, from auditory cortex to frontal cortex, especially the precentral regions of the left hemisphere. And we found that there were expressions of beta burst exactly at the rate of presentation of the sequence to which the participants needed to pay attention to. So not 3 Hz, but the 1.5 Hz inside the 3 Hz sequence. And these bursts were directed from the frontal cortex to the auditory cortex, a little bit along, what is presented here conceptually. So what I'm trying to say here is that what I'm presenting to you today is an attempt to have a holistic view of this complexity of the neurophysiological brain activity that I was showing with the traces that evolved over time. And there's a way, or at least there are testable hypotheses that we have here that may define a form of structure and architecture in space and time for the activity of the human brain. So the idea would be to collapse this into the notion of this fingerprint, which is a low-dimensional kind of summary image of what these patterns represent in a certain individual. So let me try to be more specific here. And this is everything I'm showing today is the work of Jason da Silva Castanheira, who just graduated from the lab with his PhD. He's going to move to UCL in a month, working with Steve Fleming. And so that's his work, and I'm really grateful for his efforts in that respect. And the idea of how you make brain fingerprinting work is that you need, first of all, data collection or data segments, multiple data segments or snapshots from the same individual. So we're going to call them data set 1, data set 2, not very original. They can be taken on the same day. They can be taken on multiple days. So on average, what Jason did with our seminal neurofingerprinting study is that we took snapshots of brain activity from dozens of individuals, actually almost 160. And the average duration or gap, if you will, between the two snapshots of brain activity were about 6 to 8 months, 200 days. And these were young adults. And the data is available in the Open MEG archives, which is a free open repository of MEG and T1, and also standardized testing and demographics of these participants. So we wanted to replicate what was observed with fMRI, but we had the pretension or ambition to actually push the envelope a bit further to the advantage of MEG, in the sense that because of the time resolution, we get many more data points per second than fMRI, or at least the changes you observe are way better resolved in time. And so we looked at snapshots that were 2 minutes in duration rather than 15 minutes for resting state in fMRI. Typically, it could be shorter a little bit, but not much shorter than that. So 2 minutes with MEG. And we looked at the robustness of the effects we were detecting. So the faithfulness of the fingerprint, or the stability, I should say, of the fingerprint over days, even with snapshots of 30 seconds, as short as 30 seconds. And so the pipeline is relatively basic, and that's what I like in it. There are not too many sophistications, so the simpler the better, I think. So let me try to explain to you. So we first do source modeling and source mapping onto the cortex of the individual participants from their T1 MRI using your favorite source mapping technique. And then of course, you want to define the fingerprint based on certain features or summary features of these time series. And there could be many. So first we tried with the simplest one we could think of, which is the power spectrum at each and every given region of a given parcellation of the cortex. We used first desiccant Kiliani, now we use the Schaefer 200. We don't think it's that critical as long as you have enough of those features. So just the power spectra, but also we wanted also to replicate again what our friends in fMRI did with the connectome. So we extracted a form of connectome based on amplitude envelope correlations in different frequency bands. And once you do that, you end up having a vectors of features. So P1, P2, these are the different participants, and the little genotypes, if you will, are the different values of these vectorized values of these power spectra or connectome. It could be a vectorized version of the connectome matrix, if you will, doesn't matter. But the idea is that this is the brain fingerprint of each participant. And then what you do is you cross-correlate those fingerprints that you have obtained for day 1 and you have obtained for day 2, and you are checking whether there is longitudinal stability in the fingerprint by just computing the cross-correlation between those vectors. And this is a cartoon example of what it looks like. It's a confusion or cross-correlation matrix. You hope to see the diagonal, sorry, I forgot the color scale, but the brighter the color, the higher the correlation. So you hope to see things like a very bright diagonal, meaning that each participant would correlate with themselves over time, meaning that their fingerprint is really a fingerprint and not just, you know, a snapshot of things that vary. And you want also that folks be highly differentiable, meaning that you want to have a strong diagonal element and relatively low off-diagonal elements, meaning that that person is really differentiable from the crowd. And that's an example of one person, such person. But the last one, as you can see, is more fuzzy. So this person may be highly correlated with itself, with themselves, but also quite a lot with others. And I do not mean it's a bad thing, but if brain fingerprinting is meant to be a trace of or a way to measure our similarity or difference with others in terms of brain features, the fact that we are highly versus low dimensional differentiable from the crowd, depending on who the crowd is, might be interesting and pertinent to the question. If you are a patient, maybe you don't want to stand out from the crowd of the healthy normal variants, so to speak, or you want to take a treatment that brings you back to a situation where actually, yes, you are similar to yourself, but also quite similar to healthy normal variants with same demographics. So that's the idea. And I'm just going to paraphrase what I just said. So you take a large group of folks and you actually do exactly this. And you look at the distance, a form of distance of cross-correlation of people with themselves versus others. And you take the ratio of that or a Z-score, and it gives you a score of differentiability. You can also use what we call, and others in brain fingerprinting, intra-class correlation, which is basically once you have established whether how a person is differentiable from others or not, you can actually look at a form of cross-validation by removing certain brain regions and see which are those that contribute the most to your differentiation from others. So you can be relatively specific about which brain regions are really key features of your fingerprint. And for instance, here, the brighter, again, the more salient. So we call those high ICC scored brain regions the most salient features of the brain fingerprint of a certain individual. And you see here, for example, the cingulate cortex, maybe the auditory cortex on the left, and a few other regions are more salient, more specific of that person or this group of persons with respect to controls. So that was the seminal study, which we replicated now, and I'm going to show some new results, unpublished yet. that we obtain with even bigger or more specific crowds of folks. And this is I want to pay tribute and acknowledge the huge contribution of, those who take the effort to curate and share data repositories in our community. So kudos to the Cambridge group with the Center for Aging Neuroscience with the CAMCAN database, which is fantastic. We are very lucky to collaborate also with Professor Margot Taylor at Sick Kids at the University of Toronto. She's been sharing a lot of data in children with us. The Human Connectome Project, I'm going to talk about it, especially with the twin studies. And we have our local Quebec Parkinson's Network, because now we are looking into whether this fingerprint approach can be meaningful for clinical neuroscience. And there are many others, but I wanted to really acknowledge their contribution. And I'm going to also feature, a recent tool that was developed by Bratislav Mizic, who's a young investigator at the MNI at McGill. It's a very close colleague of mine. And he was kind enough to embark me on this adventure to derive practical tools to contextualize basically the different features you might extract from MEG, for instance, like the fingerprint, but it could be the things you observe in fMRI, etc. And you want to relate those things with atlases of, T1, T2 ratio, cortical thickness, some forms of metabolic traces and markers that you get from PET, electrophysiology in the resting state, delta, theta, alpha power, some neurochemistry, the density of some neurochemistry receptors. So this was obtained by, again, a joint effort by Marcello and Justine Hansen, who are grad students with Bratislav. And we actually went and knocked at the door of many researchers who each had, a portion of this data. And they realized the tremendous effort of putting everything together, including with a toolbox that is available to us all, called the NeuroMaps Toolbox, that was just released a bit more than a year ago. So I'm going to talk about that as well a bit. So results now. So back to neurophysiological brain fingerprinting. We first look at the differentiation accuracy between these close to 200 individuals that we have. These are young adults, healthy adults, and just resting state, 2 minutes recording. And we first looked at the within session consistency of the brain fingerprints, so a few minutes apart as a benchmark. And we're going to look at broadband activity in the make sense, so from very slow frequencies to very high frequencies, but everything included, collapsed together, versus band-specific brain fingerprints, because these frequency bands, as you know, are traditionally viewed as maybe, as I was trying to explain in my conceptual slide, different roles and different functional roles. Yes, sorry, I'm trying to find the best word, and I failed. So first we look at the broadband spectrum, and so the scale goes from 0 to 100 differentiation accuracy, and you see we're getting very close to 100%. So the pink bar is with the connectome features, and the blue bar is with the spectral, just the power spectrum features. So in a sense, very similar virtually. And then we looked at from the very slow to the very fast frequency bands, and you can see that everything seems to be above 95% accuracy, regardless of whether you use the connectome versus the spectral. Actually, the color map is inverted. The blue is indeed the spectral, and the pink is the connectome. Sorry about that. And we verified actually that this differentiation accuracy was not due to environmental conditions, like the noise in the MEG room that day, that may be specific of that individual, by the way, because there is construction in the building that day. And if we look at within session accuracy, well, we're going to identify that person based on the noise in the building that day. Whereas if I go the next day and get a second participant, maybe the building is quiet, and therefore the features of the fingerprint would be driven by the noise or the artifacts. And these are the gray bars you see, which are here to control for these confounding factors. And you see that the differentiation is actually way above those ground floor levels due to environmental conditions that vary from day to day. And we verified also that movement artifacts or ECG that might still be present in the MEG or blinks did not differentiate between individuals. They know we didn't find any linear association between the fact that somebody is differentiable and how pronounced their artifact might be. So that was also very important to secure that, so to speak. And then we looked at the between session challenge, which is more exciting. So again, about 200 days apart on average, mind the scale, which is now zoomed in. It's from 75% differentiation to 100%. So with 2-minute data snapshots for each session, looking at the broadband spectrum, we were still, achieving very good accuracy at differentiating folks. And that's true for pretty much every frequency band. Again, the minimum here is 75%. With a strong, feature of the beta band, which is very dear to my heart, because again, if this is true that the beta band might be a signature of activity of internal representations, internal models and of a certain person at a given time, well, it looks like they are relatively stable, might be a signature of that person indeed, and their representations of the world. But I'm speculating here, but I'm very glad to see that in this conceptual framework I was trying to share today, the beta band seems to be very robust indeed. And then we pushed the envelope, like I said, we looked at 30 seconds snapshots of data on day 1 and day 2. And these are the cloud of points that I'm trying to show here. And you do appreciate, I hope, that of course it's more challenging, the accuracy is less, but it's still above or at least 75% for the broadband spectrum and the beta band and the gamma band, and the other bands are not as reliable to differentiate from short recordings. So I would not, have bet money on that before, and I was pleasantly surprised that, there are features that seem to be longitudinally very stable to some extent in our brain activity. And if you want to know more about stability of spectral features in the brain, I can recommend this paper by Alex Wiseman, who's a postdoc in the lab. So, and then we looked at, of course, which regions of the brain were the most salient in the fingerprint. And this is this intra-class correlation maps I was showing before. But this time we have them over all the frequency bands. And as you can see, the beta band is again, seems to be very salient, so to speak. I mean, every brain region, especially the dorsal pathway, dorsal regions and lateral temporal regions, cingulate regions, a lot of cortical activity seems to be engaged or contributing to these beta band features of the brain fingerprint. The other brain regions or frequency bands, I should say, are more scattered and less and contribute. They do contribute to the fingerprint, but in a less consistent way. Now we're going to look at whether this fingerprint is stable with aging. So yes, probably we are the same person as we age, but we know that the brain gets affected by ages in many ways. There is cortical thinning. There are many different aspects of brain activity that is also altered. So it was not such a long stretch to hypothesize that the brain fingerprint might not be stable over age, as we age, sorry. So we looked at this in more detail with the CAMCAN repository with 600 participants. We looked at the full cohort, so younger and older adults. So when I say older, I think it's up to 85 years old, but our category here is 65 and plus. But if you bring everyone together, so from 18 to 85, and that's the yellow bar, we used 4 minutes recordings of resting state, and we replicated the observations of the previous cohort with the young adults, and you see the differentiation accuracy is above 80%. And with 30-second snapshots, we are on par with what we were observing before. We are way above chance in terms of individual differentiation. Now, it's interesting to look at possible edge effects. So of course, here it's not a longitudinal study. It's a cross-sectional cohort where we look at the younger adults versus the adults and versus the older adults. And but it's hard to see any change in differentiation accuracy with age, which is a good sign, meaning that within an age group, it's not because you're older that your brain activity tends to be more variable over time. So there's this stability that still seem to be remaining at least over, a few hours, which is the duration of the session. And then we looked at whether the brain fingerprint features could help us decode some behavioral traits or personality traits. So here it's not personality, it's more like neuropsych, standard testing of fluid intelligence, which is a trait that is tested with the CAM-CAN repository. And what you see, the dots that are colored differently represent each and every individual of the 600 folks in the cohort, and the color encodes for their age. And what you find on the y-axis is the fluid intelligence score. And unfortunately, for those of us who are aging rapidly, it's not a surprise that the fluid intelligence is a bit lower than in the older folks than in the younger folks. So that's just the neuropsych data. But what was cool to see is that, so that's the x-axis. It's the true scores from reported by the CAM-CAN repository. But what Jason did is actually try to decode the fluid intelligence score from the brain fingerprint features. So he used a classifier that he trained with machine learning techniques. And you see that the decoding of the score, neuropsych score, aligned really well with the actual score that was in the database, just from the fingerprinting features. And then we looked at what changed in what differentiated the fingerprint of the older adults versus the younger adults, if anything changed, right? Because for now, we just report the fact that we can fingerprint folks from their brain activity, whether they are young or old. But if we step back a little bit, if we look at the older adults versus the young adults, what are the features that are the most salient in the young adult fingerprint versus the older adults? Because we know from electrophysiology, and we verified also from the CAM-CAN database, that even if you look actually just at the spectral features in a certain brain regions, like this one, for instance, and you look at the age effect or the effect of age on the spectrum, you see that there are features you can detect with the naked eye. So for instance, in younger adults in green, you have a greater expression of slower frequency, activity compared to older adults. And in older adults, it's kind of the contrary. You have greater expression of beta activity than than in younger adults. And we cannot see here clearly, but if I zoom, we would see that the peak of the alpha activity would actually slide to the left. It would be like a slowing of alpha activity in the older adult's brain. And because the fingerprint that we use is based on the spectrum or the frequency spectrum, I forgot to mention that after we compared the connectome fingerprint with the spectral fingerprint, and we verified that the performances were equivalent, we elected to only use the spectral brain fingerprint because it's faster to derive, and it seemed to be good enough for the things we were trying to look at, which is basically the differentiation. But this can be repeated and specified for the connectome as well. So yes, differences in the spectrum. So we would expect that the fingerprint evolve with age. And actually, that's what we found. So we looked at the saliency maps, so the ICC maps for the young folks versus the older folks, and we computed the difference. And that's what we found. We found that the most salient portions of the brain fingerprint in older adults, there's a dichotomy, as you can see. You see some pink over the dorsal pathway and some green over the ventral areas. And that actually matches the dichotomy or the differentiation between older versus younger adults. So older adults are more easily differentiable from the dorsal activity, the activity of the dorsal brain regions versus ventral regions for younger adults. And it's relatively cool to observe because if you look at cortical thinning, so we derived the cortical thinning from these participants with their T1 MRIs. And this is what you see here. With age, you see a great alignment between the topography of the cortical thinning and the topography of the salient features that are age specific in electrophysiology. So that means that there is indeed an age effect that is related to the anatomy of the participants of the structural MRI and to things that we know are affected by age in the brain structure. So that was good to kind of independently verify with a different imaging modality the alignment between we observed in neurophysiology and structural MRI. And then we used neuromaps to contextualize basically these maps and these observations. And we look at the alignment of these delta ICC maps. So again, what differentiates the old brain from the young brain, so to speak. And we found some significant alignments that could be negative or positive. So whenever it's positive, it means that a certain map, like for instance, noradrenaline cortical distribution in is aligned with the salient features of the older adult's brain fingerprint, whereas for young adults, the alignment is more with the serotonin cortical systems. So I'm just featuring this to again mention that from something that is relatively simple, which is just this power spectrum of brain activity at each cortical brain region, you can really go much deeper into some insight regarding the neurochemistry, neurobiology of the phenomena that may be at play during aging. So both from the neurochemical side of things and the neuroanatomical side of things with cortical thinning. And then we asked the question whether the brain fingerprint, a little bit like other features of our bodies, so to speak, might be heritable. And for that, we looked at the Human Connectome Project's data that features MEG recordings for singletons participants, but also dizygotic twins and monozygotic twins. So it's a very precious data set. And we did a little bit of a challenge here, meaning that, yes, we did the differentiation derivations I've talked about from individuals to individuals against the group. But also we did a matching kind of challenge that looked at whether we could match a given twin with their pair. So meaning, in plain terms, is the brain activity of a twin matches that of his brother or sister. And we hypothesized that if the brain fingerprint is somewhat heritable, we would be able to match monozygotic twins between each other and not so much dizygotic twins, and not at all, of course, singletons with twins beyond just chance or just the variations we observe in the general population. So that was the challenge that Jason took. And what he found, so there are several things to look at here. So again, this is broadband, this is delta, alpha, gamma, high gamma. It doesn't matter too much because qualitatively the results are the same. If you look at the individual dark blue bars, so this is just a replication of the general fingerprinting challenge of differentiating between people, regardless of whether they are twins or not. And yet again, you see that we are around 80%, 85% accuracy, like in the previous cohorts. And but if you look at the monozygotic matching challenge, so we looked at a given monozygotic person of a twin pair, and again, try to match them with everyone, including their twin. And actually, the twin matched much better than any other individual. So meaning that the monozygotic twins are differentiable from each other, but they match, there is a similarity between their brain activity that is way above what you observe with the rest of the population, including what you can obtain between dizygotic twins. So this is the orange bar, where you see that the differentiation accuracy is actually very close to noise levels or chance levels when you try to match a dizygotic twins with their pair, with their sibling, I should say. So that means that there is a form of heritability, right? Because monozygotic twins share most of their genomes. They differ eventually with, gene expression and environmental factors and personal history, but at least they share this genome, and therefore, most of the genome might be, their fingerprint is a signature of their similarity at the genomic level. And we verified that this was not, entirely driven by anatomical features, because of course, monozygotic twins look alike each other, including their brains, their anatomy, the curvature, the cortical thickness, etc. And so there is a trend for sure to that look at the similarity between anatomy and differentiability, but it's barely, I mean, there is a trend, but it's not statistically significant and not enough to explain the differentiation accuracy that we observe, or the matching accuracy that we observe between siblings. And another thing we did with Jason was to, again, look at the regional saliency of those brain fingerprints, just like in the previous cohort. And we looked at the heritability of neurophysiological activity. So we took the frequency spectrum in all the different bands, and we looked at how it replicates or how similar those different patterns are across siblings, whether they are monozygotic or dizygotic. And using the Falconer's method to derive heritability as a score, you end up getting those maps that you can then try to align statistically with what you observe in terms of saliency of the brain fingerprint. And what we found is that at the broadband level, but also specifically for the alpha band and the beta band again, these bands and those fingerprints features are apparently heritable and are driven to some extent by the gene pool of the participants. So Jason took that one step further and ran a partial least squares analysis between these salient brain fingerprint features that we just talked about in twins and the atlas of gene expression that you can derive from or obtain from the Allen Brain Atlas. And so by doing a partial least squares, you can look at the shared variance between two things that are apparently completely different. So the different fingerprint features from different brain regions, different frequency bands, and how many different genes are expressed in the same regions of interest, or at least those that are available from the Allen Brain Atlas. And by running the PLS analysis, you can see whether there is a possible alignment between those two things, or correspondence, shared variance between the two. And actually, Jason found that there was a major, latent variable, as we call it in PLS, that explains the shared variance between the two features, with, again, a very typical dorsal versus ventral dichotomy, that this is for the latent variable from the genes expression, and the same dichotomy was found for the fingerprint features with the saliency maps I was talking about. So you see a very clear alignment between the two. And then you can look at basically resources that are, again, available to everyone through the ShinyGo pipeline, which is a gene ontology pipeline, and the Geo database. And you can look at how these different features relate to more intimate mechanisms and aspects of neurobiology and neurophysiology, and to, again, contextualize your findings based on simply brain fingerprinting individuals. I have to say, this is not specific of brain fingerprinting. You can look at any given map from your own studies. And if you want to contextualize those maps in a certain population versus another, for instance, you can use these tools the same way to, again, provide some more insight from neurobiology onto your observations with non-invasive recordings. I think I should skip and get into the last portion of the talk. It's only one slide before I wrap it up. So as I said, I'm particularly interested in looking at how the fingerprinting device, if you will, can be used as a tool to maybe get better insight into the mechanisms of disease and also diagnostic, So we were lucky to access, again, the Quebec Parkinson's Network data, which consists of a deep phenotype of about 80 patients, including MEG resting state, T1 MRI, and many clinical factors. And we replicated basically the observations. Again, it's important to be able to replicate independently with the same pipeline our observations of differentiation between individuals. And for that, we looked at age-matched healthy controls from the CAN-CAN, even though they use a different scanner from the one we use for the Parkinson's data, so CTF versus McGill. And also the PreventAD, which is yet another cohort of healthy participants that we used as age-matched controls for the QPN, the patients. And we looked at differentiation between controls, replicated the observations, the same as before. And we noted that for Parkinson's, the differentiation accuracy was still quite good, but degraded with respect to controls. And so we wondered what could be the reason for this degradation in accuracy differentiation or differentiation accuracy. We checked whether they moved more during the scan, and that was not the case. And we could not identify obvious artifacts, non-brain artifacts that could explain this little drop, but yet significant drop in terms of differentiation. So we will talk about spectra today in neurophysiology. You probably have heard about, FOOF, spec param, arrhythmic versus rhythmic features, decomposition and parameterization of the spectrum. And that's exactly what we did. We looked at the how the arrhythmic components would help differentiate between people. So instead of taking the full spectrum, you take only the 1/f background activity and take that as a feature in every brain region for the brain fingerprint and do the same thing again, the differentiation between individuals. And as you can see, by looking at the arrhythmic activity, we saw some reduction in the ability to differentiate between people, and especially in Parkinson's patients. Whereas if we kept only the rhythmic portion of the fingerprint, then we went back to very high levels of differentiation between individuals, including between patients and controls. So it looks like what it means is that, I think I have a figure for that. No. It means that the rhythmic components of the spectrum are more stable over time in a given individual, including in a Parkinson's patient, and therefore are more reliable to derive brain fingerprinting features, whereas arrhythmic background brain activity fluctuates more substantially over time. And therefore, if you take two snapshots of just arrhythmic activity, so to speak, in the same person, they tend to look less alike than the rhythmic counterpart. And this thing has been observed also with fMRI, to the best of my knowledge, where actually brain fingerprinting or actually measurements of local fluctuations over time of the characteristics of the bolt signal seem to be more pronounced in Parkinson's patients, but also other types of patients. And this is maybe what we capture with the arrhythmic feature here. And the rhythmic feature seems to be more stable again. And so we took the PreventAD sample as a control and then the CAN-CAN sample as a control just to replicate our observations, because we found somewhat a puzzling observation that the saliency map of the Parkinson's brain fingerprint seemed to point at multiple regions, but essentially the right somatosensory cortex. But this is also what we observed with the CAN-CAN dataset. So this seems to be something very strong there and very specific of our Parkinson's patients, at least the cohort we've been looking at. And indeed, when you look at this power spectrum, just the rhythmic component in that region, you can see a very clear differentiation between patients, healthy controls, and even among patients you see you might be able to differentiate between disease stage. So in terms of motor symptoms, we use the Horn-Yahr stage clinical staging scale. And you see that there is a clear differentiation again between early versus late or advanced, I should say, disease stages in that region. So that made sense, maybe, that we look at this as a very salient differentiating feature of the brain fingerprint because it reflects disease staging in our cohort of PD patients. So what we did is actually try a little bit like fluid intelligence. Instead of decoding fluid intelligence scoring, we detected the motor symptom scoring and disease stage just from the fingerprint of each and every region. And that's what we found. This is the decoding accuracy of the Horn-Yahr score. And you do find indeed, you confirm that this sensory motor region, especially to the right, but to some extent to the left as well, but more pronounced to the right, is very strong and very reliable in PD patients. So there is meaningful information, clinically relevant information that you can extract from this data. I will skip. So why should we care? I'm going to conclude here. Well, one thing I dreamed of some years ago was to actually look at leverage big data, possibilities to actually look at how a certain individual data points person would deviate from healthy normative controls for the same demographic with the same demographic variables, etc. And we used a little bit of that in epilepsy, where basically we looked at a population of healthy controls, a given individual patient, and for each patient, each voxel, we looked at where their brain activity would sit in the distribution of normative controls. If it's more in the tail, you would actually paint that voxel a certain color, and you proceed with other features. And at the end of the day, you have a summary figure that looks a little bit like this. But with fingerprinting, you make it more systematic. And eventually, what you would like to do is look at these patients like this blue dot here and see how far it is from their peers, basically. And of course, you want to anticipate a treatment or an intervention that would bring that person as close as possible back to their peers and healthy peers. And that's where we are trying to get now with the rest of the story. We are trying to look at how we can, first of all, measure this form of distance in a low dimensional space of brain activity with fingerprinting, and how we can derive interventions, if possible, or at least measure the efficacy of a given treatment, given drug therapy, and see how it normalizes in a good way brain activity. With that, I want to thank my team, Jason in particular, but everyone also in the group for such a dynamic, creating such a dynamic and inspiring environment for me. And I want to thank you very much again for sticking around and for your kind attention. Thank you.